Good morning. Welcome to today's webinar. Today's webinar is on the subject of protein loop modeling. There's this recording as well as the other webinars in this series and previous ones from last year and years before can be found on our YouTube channel. If you just search MoleSoft, you'll find it here. So today's webinar topics are including how to model a loop region using a knowledge-based approach, so using a database of loops that's derived from the PDB, and then also how to model a loop region sort of in an ab initio way using a ICM bias probability Monte Carlo optimization method. Then we'll be moving on to protein engineering, how to design a new loop, how to graft a loop from one protein to another. So if you have a loop that you want to move from one to another, you can graft it. Also, if you have a loop region and you want to investigate what residues are preferred at certain positions in the loop, you can use this option. And we'll also look for loop regions that have a similar conformation to that in your model. So there's different approaches to loop modeling all built into ICM. This is one of the webinar series that we're in at the moment is modeling. So we started off with homology modeling. Last week was template-based modeling. Today's loop modeling. And then we'll finish off with predicting the effects of a mutation in ICM-Pro and also how to model a binding pocket using known ligand information. That's in a couple of weeks' time. Read this book that myself and Ruben Abagayan, who is our company founder, we edited it back, and it contains useful chapters on everything we've covered today. I'll be happy to send you PDFs of some of these chapters if you get in contact. Just a quick slide about MoleSoft. Ruben is our company founder, Max Tochoff is our principal scientist, and Eugene is our principal developer. We're based in San Diego in California. And if you're wondering what ICM stands for, it stands for Internal Coordinate Mechanics. We don't use Cartesian coordinates, we use internal coordinates. And you can read more about this method as well as the bias probability Monte Carlo method in these papers here. So with this webinar series, you can download ICM and you can follow along during the webinar or after the webinar. We can provide a license key for you. If you haven't already done so, go to molesoft.com/support, use your company or university email address to log in, and then download ICM-Pro full package, and you'll see a 12-character host ID. Just email that to me and I'll send you a license key. And ICM-Pro runs on Windows, Mac, and Linux. So today we're concentrating on loop modeling. And there's different approaches which we'll talk about. And this is just an example of a success story with triose phosphate isomerase. Basically protein engineering experiments were carried out using monomeric triose phosphate isomerase. And this loop 1 in the monomeric form is very disordered in all the crystal structures. And the idea was to, this is work done by Ruben Abagayan and the Varenga group. And the idea was to try and stabilize that loop using different modeling methods inside of ICM. The aim was really to try and make this loop rigid because in the dimeric form it is rigid because it's forming an interaction with the dimer. So ICM modeling suggested that the loop should be maybe one residue shorter. And then they solved the crystal structure of that loop region and shows that the experimental loop agrees with the modeled loop structure, and the direct superposition was about an RMSD difference of 0.5. So it's a good example of using protein engineering for using ICM for protein engineering. So basically for loop modeling, there's two different approaches. You can search a database of loop conformations, or you can use Multisoft's full atom ICM stochastic global optimization procedure. So this is a slide taken from Max Totroff, our principal scientist, his chapter in this modeling methods in molecular biology book. And it shows some different approaches that you can use in ICM for modeling. So one approach is to use the knowledge-based loop modeling approach. So this uses a database of experimentally observed polypeptide chain conformations extracted from the PDB. And these are loop segments. These are segments of loop regions, and they geometrically match the terminal residue positions of your loop gap. And then the loops are scored according to their best fit with the rest of the structure. The other approach would be which is like an ab initio method, and so it's based on sampling. So it's, rather than looking for loop regions from the PDB, you're sampling the loop using the force field and the bias probability Monte Carlo method. Although knowledge-based loop modeling methods are generally faster, they are obviously limited by the available amount of experimental data in the PDB. So, but whereas the ab initio approach in principle can predict novel structures that aren't observed in the PDB. So the idea is to find the native conformation of the loop, which represents the global minimum of its free energy. So ab initio methods identify the near-native structures via global optimization, and the success of the ab initio method depends on two main factors: the ability of the conformational search algorithm to locate the lowest energy minima, and then also the scoring function as well. So you need to be able to rank near-native solutions of your loops. So both these approaches are in ICM. Just for time I'll show you how to run them in ICM. But just for time constraint, we'll be showing the knowledge-based approach, because it's quicker, obviously, than the sampling. But the reality is that we usually in this is like an ideal case. You know exactly where the C and N terminal is. But in some modeling cases, most modeling cases, there's ambiguities about the exact position of the C terminal and the N terminal of your loop. So it's kind of an inexact environment. So this is more a realistic starting point. So you need some way of modeling the loop as well as the region around the terminal of your C and N region. So you want to be able to incorporate some flexibility here and here when you're modeling. So that's all included in the ab initio method that we have. So loop modeling accuracy, there's some good comparisons on published. I'm not going to show them against other, but ICM performs very well compared to other methods. But generally speaking, when you get, so this is the loop length, the accuracy is pretty reasonable for like 6 residue loops, 7 residue loops, and obviously it tails off the longer the loop region is. This figure is taken from Max Totroff and Ruben Abagayan's paper on the ICM force field. One of the applications was on loop modeling, and so there's a 7 residue loop here. Green is the crystal structure, orange is the predicted, and so it's very good. Some other where the loop region is not well predicted is because you need to make sure that you have the full protein structure. So for example, this loop, if you modeled it in reality in the crystal structure it's interacting with another subunit, so you need to make sure you include all the data in order to achieve a good prediction. Okay, so if you're using the knowledge-based approach, you need to make sure that you download the latest ICM loop database, which is derived from the PDB. When you download ICM from our website, it does include a small loop database, but it's not the full database just due to size constraints. So to download it, you'd go to the help menu, and then you choose MuleSoft add-ons, and then there's a link there to download a file called full.lps. And then you would save that into your ICM directory. So for example, in Windows, that would be C drive program files ICM-Pro. In Linux, it'll be your ICM home directory. So we're going to go through an example of loop modeling in ICM using the knowledge-based approach, but also show how to do it with the sampling. So it's an example from the PDB. It's a crystal structure of the anthrax protective antigen, which is the cell binding part of the anthrax toxin. And the anthrax toxin is composed of this protective antigen as well as lethal factor and the edema factor. And the protective antigen translocates the lethal factor and edema factor into the cell. So, but it's an interesting example because if you read in there's two crystal structures. There's one here, a lower resolution, and a higher resolution structure. And in this loop region, there's a proline, and the higher resolution structure actually shows it as a cis proline, whereas in the lower resolution, they've modeled it probably into the loop as trans. So you would maybe a bit suspicious of this, but if you run the full simulation of this loop region, you do actually find that the most energetically favorable conformation of this proline is actually in the cis conformation. So how do we do that practically in ICM? First we need to load ICM. So you should have an icon here, and I have already read in the two PDB files. So the two PDB files have accession number, the PDB code 1ACC and 1TZO. So just going to zoom in. So you could read them in, go to search menu and type in one. You can read them 1ACC and 1TZO. So now you have them displayed. We can see that. Let's just toggle them on and off. So the low resolution structure, this proline, you can see here in trans. In the high resolution 2.1, this proline. So in this example, we're going to remodel that loop and see which one is the most energetically favorable. And so to do that, to run any modeling in ICM, you really need to convert your X-ray structure into an ICM object. So to do that, I'm just going to delete. We're only interested in the A chain, so I'm just going to delete the other chains. So you can select them, right-click, and choose Delete. So to convert it to an ICM object, we right-click on the menu here, on the name of the protein you want to convert, choose Convert PDB, and then we have the options here, which we discussed in previous webinars. If you're doing this properly, you would optimize all hydrogens, You have subjective decisions. You have decisions to make about whether to keep certain waters or delete all waters, or whatever. But I was going to choose replace the original, overwrite this X-ray structure with the ICM object. So it's going to also optimize certain residues that are the orientation are ambiguous in the X-ray in the maps. So just go OK, and this will convert to an ICM object, and then you can use it to model. Okay, so it's now converted. So now you see here it says ICM rather than X-ray shown here. So we know this is an ICM object. And the loop we want to model, so I'm just going to undisplay the high resolution structure. So the loop we want to model is from residue 4, 4,1, let me just double. It's this region here, 410 to 414. So you can select this region. You can see that it's blue here in the ICM workspace where I've made the selection, 414. It's a small part of the loop, just for speed, but we can display that region. So to model this loop, we would go to Mole Mechanics, choose the option Loop, and then there's this option Sampling Modeling here. So if we click that button, it gives us a dialog box shown here. And you see at the top here, there's two tabs. This is interactive. Interactive is the knowledge base. It's going to search the loop database. Or you can choose high precision sampling, which is the full optimization of the loop, independent of any template from the PDB. So in the real life scenario, you probably, if you want to quickly find a set of loops, you can choose interactive. But if you wanted to check the energy of this loop and this proline, which conformation of this proline is more preferred, you would definitely have to use the high precision sampling in batch approach. It's asking you for the loop residues. We've selected them. The selection is propagated to the 3D. You can see it says 4 residues. The effort is the length of the simulation that you'd run. And 1 would be reasonable for this kind of loop. Maybe increase it slightly if you've got a very long, over more than 8 residues, 9 residues, you may want to double this effort, but this would be should be fine. Well, it is fine for this example. You can also sample surrounding side chains. So the loop will be sampling the conformations as well as the side chains as well. So you can include that. Truncate remote parts will just chop out this region of your protein. It's up to you if you want to do that. And then use original conformation will keep this original conformation when it starts the simulation. So it will start from that position. So that's high precision. So when you run that, you will see up here simulation that a background job is running. You'll see a progress bar here. Depending on the size of the loop, it could take an hour to 3 or 4 hours or something you'd leave overnight to run on one machine. So it's quite a long simulation. The interactive approach, whereby we go to the PDB, our loop database, this is what we're going to do today. So we're going to choose interactive. Once again, it's asking us for the loop residues, the four residues. We're going to do a loop database search. And you have 2 options: store in the object. This will store all the loop conformations that are favorable for that distance in the PDB in our object, or we can have a table of the different objects. But I'm going to choose store in object. So store a stack, what we call a stack, ranked by energy, by prediction, the most favorable loop in the ICM object. So just go OK, and we should start seeing it in a minute. It takes about a minute to run, or 2 minutes to run. So it's checking the loop database for loops with similar distance and sequence that are already in the PDB. It's found 1,840 confirmations so far, but it's compressed those into a set of 13 confirmations which we can look at. So once it's run, you'll see that in the ICM workspace we have so one TZO is the low resolution structure we're modeling on. We have a stack we have this option here called stack, and it tells us that there are 13 confirmations of that loop in this stack. So there's two buttons here. One, if we press play, it will play a movie of the different confirmations that it's found for that loop. To stop that movie, you need to press the red button here or press escape. The other option is this this gray play button that will enable you to toggle through the different confirmations that it found. And if you click on this plus button, that will show you these the loops ranked by how well they fit. So the first one is 1. You can toggle through them here. If you want to keep one, for example, if you wanted to keep this as a separate object so you can compare, you can right click excuse me. So you can actually just display that, and then you can just clone this. So right click here and choose clone, and that will have you if you say clone, go OK, and you can say conf5 or something like that. So now we have another object here. So if we can take this one back to 1, we can easily compare conf5 to confirmation 1. And because it's actually searching the PDB database, and this PDB is actually in our database, it does find the sys even without the global optimization simulation. It obviously finds that loop from one TZO and it maps it on. So it's but obviously in a module model example, you're not going to find the exact match. So this is a way to toggle through your different loop confirmations. Okay. Go back to the slides. So you may also be interested in loop design. So you may have a gap in your loop, or you may have a region where you want to do some protein engineering and find the preferred loop region there. So to do that, you need to use the option Mole Mechanics Design Loop. And the option will search for loops, again, in the PDB, which have a similar gap as we were doing earlier, and similar distance and geometry, and it will output energetically favorable solutions, and it will tell you which PDB it came from. So, and it will report the energy of that loop, the RMSD between the loop it's placed in there and the loop it took it from the crystal structure. And it will tell you the residue numbers from that PDB structure where it took that loop from, and the resolution of the structure. Okay, so let's show that practically in ICM. It's going to close. It's going to open a new instance of ICM. Go File, and okay. So this is a kinase. So we color it, we go to display, color by N to C. We can clearly see the N terminal here and the C terminal here. We have this hinge region, and there is a gap here between alpha C and this beta 3. So if we wanted to find a loop, design a loop that would fit here, we do the following. We can go to mole mechanics, go loop, and then choose the option design. So it's mole mechanics loop and this option design. Okay, and that brings you this data entry box. So we need to tell ICM which residue is the N terminal of your loop that you're trying to design, which residue is the C terminal, and then you want to tell ICM what kind of loop you want to design. So how long it's going to be, minimum length of 2, maximum length, I'm going to put 6 for the maximum length here. So it's a big gap. So you put 6. Okay, so the N terminal, we make a selection. It's got to remember which side is, I think this is the N terminal, this is the C terminal. So the N terminal, we select it, and it's looking for this orange cross, so it's looking for an orange selection. If you can see, it's orange and green. So we have this other option here called green to orange selection, so we click there, and you can see that N terminal is orange, and then the C terminal is this one, 411. So we have an orange selection for the N terminal and a green selection for the C terminal of our loop, of our gap, and I use the, just for convenience, I use the selection tool here, which selects residues in the ICM workspace, in the ICM GUI. And so you just go, you choose minimum length of your loop, maximum length, and you go OK, and it will start looking for loops in the PDB that match this gap, that are energetically favorable in this region. So then you will get a table, and it only actually found, it found 5 different loops that match, that have good energy and RMSD. So you can toggle through those. It's going to show you. In the table, you can go through each one, just double click, and it will load that loop designed in. And it also has a plot here of energy versus the length. So the most energetically favorable one only had two residue. This one's shown here. There's some with 3, and so you can use this plot to find which one. So it tells you the RMSD between the one it's modeled and the crystal structure. It shows you where it took it from PDB 1TYU. If you zoom in, it's the PDB structure it took that loop from, and also the residue numbering shown here, and resolution and length. These are the variables of the loop. You could probably use this to, if you have many, in some cases you may get many solutions, so you can use this to cluster and choose, you can cluster this and find diverse loops based on the variables of the loop region. So this is loop modeling. So you may also, when you're modeling a loop, you may want, if you're trying to engineer a new one, design a new loop, you may be interested to find out which residues in the loop are preferred. So for a given loop region, this method scans the PDB database for segments that have similar conformation and then calculates the relative frequency of amino acid residues at each position in your loop. So this is a 5 residue loop, and these are the amino acids. It's obviously longer than this, cut it off at arginine, but you can see at each position which residue is preferred based on the PDB. So for example, in beta turns, depending on the type, a glycine or a proline might be preferred, so you can use it for design. So let's look at an example. I'm going to open a new ICM. So all I've done here is read in the PDB file 1xbb, converted it to an ICM object. It's you can see it's ICM. Okay, and it's a kinase again, and we can find the preferred. I've actually already run it, so I can hide that. So you can find the preferred residues for a loop. So first you need to select the loop. So you may have already modeled the loop, and you can use it from there. So I'm going to select residues 380 to 384. This is a very tip of this glycine-rich loop on this kinase. So to run it, you go to mole mechanics again, choose the loop, and then choose the option preferred residues. And there's sort of an RMSD comparison. This value, the default value is fine. Just leave it the same. It's comparing. It's looking at the angular RMSD compared to the ones it finds in the PDB loop database, and just go OK. So the default value is fine. And then you will get a table called loop residues preferences. So you can see that R1 is the glycine, the first glycine. You can see that glycine actually is in that position is relatively frequent, but the glycine is also in the 4th residue in that loop, and that's also a very common region. So you can see that in all the other, it's the most highest predicted residue for that region, and that 4th residue for that. So it's the only one actually that gets a hit. So this is a way to, if you're trying to design a loop and trying to think which residue to place at each point, this is a helpful guide to do that. Another useful tool is called Find PDB Segments. So this also not only compares the residues, but it also compares the conformation. So it will find loops in the PDB that have similar conformation to the loop that you are looking for. So to run that, you run it the same way as before. So if we're looking for other structures with similar loop, this similar glycine-rich loop here, we make the selection. We go to mole mechanics. We choose loop, and we choose find PDB segments. And it's going to so the loop residues selection is shown here. The RMSD, we can play around with that if you wanted to. If you wanted to find the more diverse other structures, but this is going to find similar ones. And maximum number of hits returned is 500. So just go OK. And so we get a new table called loop PDB segments, and these show loop regions which have similar conformation rather than sequence. Sequences are different, but has similar conformation. So it has the it lists the PDB code and the residue where that loop starts, and the RMSD, angular RMSD, which is slightly different from regular RMSD. So so obviously the smaller the better match, shown here. So you could read in that, and the A stands for the A chain. So this is you'd load the A chain of this PDB to find that loop at this point, and find similar confirmation. So these are all methods you can use to help in your design. Okay, we're going to end with how to manually, so it's more modeling, this one. So it's how to manually graft or copy a loop from one structure to another. So this is putting more into protein modeling. So if you have a loop region, so you may have found, using the other methods we discussed, you may have found a loop region that has similar conformation, and you want to move it from that structure to your structure. So here, the red structure has a gap. You can, in this loop region here, you can see these dashes mean there's a gap in the chain, and the green structure is the loop that we want to transfer to the red one. Okay. So practically to do that in ICM, we need to read in the two crystal structures. So here we have 1XBB, which we were using earlier, and for FL1. If we extract the sequence from each one, we can make an alignment so we can see where the gap is that we want to, the loop we want to transfer. So just make an alignment. You make the alignment by, you can drag and drop this sequence into this one, it'll make an alignment, or you can select them and then right click align sequences here. This is just so we can see where the loop is that we're going to model. Okay, so the loop of interest is this one. You can see in the alignment that the loop is in 4FL1 at the top, and we have a gap here. So we're going to model this onto here. Okay, so if we select this region in the alignment, the selection is propagated to the structure, and then we can clearly see where that loop is. So if I just color this as in the slide, if we color this red and this one is green. Okay. So we're going to move, we've done all the analysis using mole mechanics, loop, find PDB segment, and we're going to move this segment to our model, to our structure, our model, which is the red one. So to do that in ICM, we go to mole mechanics, go to loop, and then we choose the option graft. It's asking which is the N terminal, so we have 528 and 533. So the N terminal is 528, residue 528. If you right click on a residue, you can see what residue you're clicking on. So this is, it shows here, this is residue 528. So in this dialog box, the way it works is you right click here and you left click on that description here, and it propagates that information to the N terminal here, to this dialog box here. And then we do the same with the C terminal. So we right click on the atom, and then we left click here, and that will place the definition of that residue into that terminal box there. So right click. So now we have the N terminal is 528, C terminal is 533. The next step you need to, I forgot to say, is you need to superimpose your structures or superimpose the loop region. So to superimpose the loop region, you can just select the region, and then you go to Display tab here, and then you use Superimpose button, which is this one here. So you select what you want to superimpose. So I'm just selecting, you could select in the alignment, for example, and then click on Superimpose, and that will superimpose that loop region. If you want to do other superposition, you go to Tools, Superposition, and then you can be more specific about what you want to superimpose. But this this superimpose button here does a good job. So you select that loop, superimpose. So now it's ready to. This is quite important because the N terminal and the C terminal need to be in a reasonable vicinity in order to make that transfer of that loop region. So I lost the. I had to place these back in. And the loop, so it's asking you for where you're going to graft the loop to. You're going to graft it to this residue where we have a gap in the on the N terminal side, and we're going to graft it to this residue on the C terminal side. And it's asking us which loop do you want to graft, and so the answer is here. We can select it in the alignment, or you can select it graphically. So you need to select the terminal residues shown here. So matching the terminus of each one is quite important. So ICM can try again to get the best fit. You can assess before you run it how good the N terminal and C terminal match between the structure you're taking the loop from to your model where there's a gap, and you can assess it just by clicking this button here. If we click that, it tells us the RMSD value is 0.7, and it gives a rating of how good the terminal matches are, and it's fair. So we'll get a good model with fair. If it says poor here, you need to go back and check the superposition of the N and C terminal and see if you can improve this RMSD in order to graft it properly. So then you just go OK, and it will graft that loop region into that gap, and you have a new structure. So if we display this loop again, it shows in yellow that loop that it transferred from this structure. Okay. So in the previous webinars, we covered homology modeling, and in the full model builder, if you go back to the webinars online, you can check that ICM, when you build a homology model, it does all this loop modeling on the fly in the background using full refinement. But obviously there are certain cases where you may need to remodel a loop, or you may want multiple confirmations of a loop in the binding pocket. So this is when these options are helpful. So that covers basically all the tools for loop modeling in ICM. I want to thank you for your questions. I'm sorry I didn't get to them during the webinar, but I'll try to get them afterwards. Next week we are covering how to project the effect of a mutation on protein stability, on ligand binding, on peptide binding, and on protein-protein interaction sites. And then the week after that we do ligand-guided modeling, where if you have a known ligand that binds, you can use that model to that ligand to mold your binding pocket, to open up the binding pocket and build multiple representations of the binding pocket for drug design. Okay, so thank you very much. I'm going to